Good morning, students. In this video we are going to discuss about ribosomes, cytoskeleton, cilia and flagella, centrosome and centriole from the chapter Cell, the Unit of Life. In previous video we had a discussion regarding some cell organelles, and in this video we are going to discuss about these cell organelles which can be seen in prokaryotic cells and eukaryotic cells. Firstly, we are going to discuss about ribosomes. Please see the screen. On the screen you can able to see the ribosomes diagram. Now let's enter into in detailed explanation. Ribosomes are the granular structures first observed under electron microscope as a dense particles by George Pallade in the year of 1953. Students, as we know, in previous class we had a discussion that electron microscope excavated and elaborated deeply in the decade of 1950s only, and after 1950s electron microscope explanation only, all the cell organelles detailed study made very possible. So George Pallade, with the help of electron microscope, discovered dense granular particles in the cytoplasm of the cells, named to them as ribosomes, and discovered in the year of 1953. They are composed of ribonucleic acid, shortly called as RNA, and some proteins, and they are not surrounded by any kind of membrane, since they are called as non-membrane bound cell organelles. Okay, ribosomes are non-membrane bound cell organelles in both prokaryotic cells and as well as eukaryotic cells. Next, let us see one more important point regarding ribosomes. The eukaryotic ribosomes are possessing subunits, and prokaryotic ribosomes are also having subunits. As we discussed, every ribosome has two subunits. Eukaryotic cells like sperm cell, bone cell, muscle cell, nerve cell, etc. possessing ribosomes, they are 80S, and whereas the prokaryotic cells, bacterium possesses ribosomes, they are 70S type. So S stands for sedimentation coefficient, Svedberg units. So here I would like to tell you one important information regarding these subunits. And 80S type of ribosomes are consisting subunits such as 40S and 60S. So the smallest subunit is 40S and the largest subunit is 60S. In eukaryotic cells like muscle cell, bone cells, and sperm cell, etc. Together as a complete ribosome, they are present, known as 80S type of ribosomes. Understand? Next, let us talk about prokaryotic cells, ribosomes, and their subunits. Prokaryotic cells like bacterium possesses two subunits. One is smaller, one is larger. The smaller subunit is 30S and the larger subunit is 50S. Together present as a one single ribosome called it is nothing but 70S type of ribosome. You can see on the screen in detailed. And what is the use of the particular unit Svedberg or sedimentation coefficient in ribosomes? The unit helps to indirectly measure the density and size of the ribosomes. Density and size of the ribosomes both in plant cells and animal cells. And both 70S and 80S type of ribosomes are possessing two subunits as we discussed. These ribosomes concept is important for short answer for 3-mark question for board examinations. Hope you understood. Now I am going to another important cell organelle. It is nothing but cytoskeleton. Cytoskeleton structure you can see on the screen in both plant cells and animal cells. It is elaborate network-like structures, and they are just like filamentous and made up of with proteins. So there are filamentous proteinous structures present in the cytoplasm of both the eukaryotic cells and prokaryotic cells, and these filamentous proteinous structures together collectively referred to as cytoskeleton. Okay, the cytoskeleton in a cell are involved in a specific functions. You can see the cytoskeleton diagram on the screen. It plays variant functions in both the plant cells and animal cells. There are many in number. Cytoskeleton involved mainly in some few functions. Let us see what are those. Such as mechanical support to the cell and motility of the cell and the maintenance of the shape of the cell. Remember, cell shape is giving by the cell wall in plant cells, cell membrane or plasma membrane in animal cells, but that particular shape is maintaining by the cytoskeleton spread throughout the cell and giving maintaining perfect shape by this cytoskeleton only. You can see the cytoskeleton on the screen. Okay, so now I'm going to another important 5 marks category concept about one cell organelle, cilia and flagella. Cilia and flagella are very important locomotory organs in both prokaryotic cells and eukaryotic cells. Cilia, it is a plural, singular for this cilium. Flagella is a plural and singular for this is flagellum. They are just like hair-like outgrowths of the cell membrane. You can see from the cell membrane they are projecting outside as a hair-like structures, both in prokaryotic cells and eukaryotic cells. Example for prokaryotic cell, bacterium. Example for eukaryotic cells, we can take paramecium and other rest of the cells also which possessing locomotor organs. Let us go for some more information regarding cilia and flagella. Cilia are small structures which work like votes together, causing the movement of the entire cell in the water and even the surrounding fluid. It means water also can be moved with the help of cilia or flagella. So either the water can be pushed back by the cilia and flagella to move the body ahead or forward, or the entire organism also can move with the help of this cilia and flagella. In such a way they helps in locomotion. And one interesting point regarding flagella are comparatively longer and responsible for cell movement. And the prokaryotic bacteria also possesses flagella, but these are structurally different from the eukaryotic cells flagella. Understand? So in prokaryotic cells the flagella structurally different, and it is quite different with the flagella in eukaryotic cells. Now I am going to explain the structure of cilia and flagella. Structure explanation as follows. The electron microscope study of cilium or flagellum shows that they are covered with the plasma membrane. Students, as we discussed just now, please watch the screen. There are two organisms, for example, Chlamydomonas, which is having flagella, cilia, which is having paramecium, are present on the screen. Let us see in both the organisms how locomotion takes place, how the structure of cilia and flagella are present internally. And electron microscope, which came into usage in the decade of 1950s, made the scientific study very easily. Before 1950s, it was unknown by people, it is unknown to society what internally present in every cell organelle, how the internal structure is present in detail. So after electron microscope usage only, after 1950s only, the elaborate explanation of very minute cell organelles came from dark to light. Example, as we discussed, ribosomes and even now cilia and flagella. And cilia and flagella are emerging from the plasma membrane. This is very important concept for NEET examination. Please write down. So what is, from which cell organelle, from which location cilia and flagella are emerging? Plasma membrane, not from cell wall. Make sure this point must be clear to everybody. Next, their core, it means internal structure or internally. Their core called the axoneme, and this axoneme possesses a number of microtubules running parallel to the long axis. As we seen, flagella is long in structure comparatively cilia, right? So throughout the long structure of flagella, these microtubules are running parallelly in the core or central region of the cilia and flagella called as axoneme. Understood? Next, the axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules. So 9 pairs of doublets. Doublet means 2, right? So total 18 microtubules, but they are present just like a doublets since 9 in number. So 9 pairs of doublets are radially arranged peripheral microtubules. So these 18 microtubules are present as a 9 pairs of doublets. They are situated at the border side, also called as peripheral side. You can see on the screen very clearly. So 9 microtubule doublets are present in the peripheral side. Understand? Please watch the screen. In this diagram you can see very clearly. Next, one pair of centrally located microtubule. You can see one pair centrally presented. So such an arrangement of axonemal microtubules is referred to as the 9+2 array. This is very important for 1 marks category. What is meant by 9+2 array arrangement of microtubules in cilia and flagella? 9 refers to 9 peripheral side microtubule doublets, you can see on the screen, and 2 refers to, it means one pair of microtubule doublets are present in the centrally, but they are maintaining gap. They are not as a pair, they are individually present in the center, so they are considered as a 2 only, not as a pair. So 9+2 arrangement of microtubules can be seen in the cilia and flagellum internal structure or transverse section, which you are seeing on the screen right now. It is called as 9+2 array. Have you understood? Now one more structure explanation. The central tubules are connected by bridges. So central, how many tubules are there? 2 tubules are there, right? They are connected by a bridge. See, you can see the bridge green color, which is present in the center of the cilium and flagellum transverse section. And this bridge is, these 2 microtubules are enclosed by a sheath called as central sheath. The central sheath is here in red color, you can see on the screen. Understand? So that is central sheath which is covering or enclosing the 2 microtubules which are present in the center, just attached or connected with a central bridge. Understand? Next, which is connected to one of the tubules of each peripheral doublets by a radial spoke. You can see radial spoke here, which is in light green or radium color. Those are called as radial spokes. So from the peripheral doublet, from one of the microtubule, one connector is emerging towards the center region, towards the core region, called as radial spoke. So what is the role of radial spoke, which they are appearing in a radium color in the diagram? They are a connector. So with whom they are connecting? Microtubule which are present peripherally, it means their 9 doublets are connected with one of connecting by one of the radial spoke. Next, thus there are 9 radial spokes are present. So from each doublet, how many spokes are emerging? 9. So 9 doublets and 9 radial spokes are present. From the doublet, from any one of the microtubule, radial spoke is emerging and presenting towards the central sheath. Next, the peripheral doublets are also interconnected by linkers. So students, here I will show one more diagram. You can see in this diagram, students, the pink color, those are called as linkers. So the peripheral 9 doublets are also interconnected by linkers. So how many linkers can be seen? 9 linkers. 9 peripheral doublets, 9 radial spokes and 9 linkers. Understand? So one more important point, both the cilia and flagellum emerging from the plasma membrane only, but they emerge from centriole like structure called basal bodies. In previous diagram we have seen the basal bodies. So both the cilium and flagellum are emerging from centriole like structure called the basal bodies. This is important for NEET examination. Understood? Students, hope you understood the cilia and flagellum structure explanation. Now I am going to explain one more important cell organelle. It is centrosome along with centriole. First, let us talk about centrosome. Centrosome is an organelle usually containing two cylindrical structures called centrioles. You can see on the screen centrosome structure, which is spherical or round in shape. In this centrosome there are two centrioles. Understood? So they are cylindrical in shape. Both the centrioles are cylindrical in shape. They are surrounded by amorphous pericentriolar material. It means one colorless or shapeless material can be seen on the surface of the centrioles, on both the centrioles. That's the specific characteristic of the centrioles. On the centrioles, a shapeless or colorless material covered. Understand? So both the centrioles in a centrosome lie perpendicular to each other. So perpendicular to each other means you can see here in this 90 degrees can be seen. So one is vertically present, one centriole is horizontally present. Together they are present in the centrosome, but the arrangement of centrioles in a centrosome is perpendicular to each other. So 90 degrees can be seen between these two centrioles. And each centriole perpendicular to each other, in which each has an organization like the cart wheel. So the organization is just like a cart wheel. Understand? So here let us see structure explanation of centriole. Centrioles are made up of 9 evenly spaced peripheral fibrils of tubulin protein. You can count here on the screen, very clearly visible. They are in green color, triplets. In previous diagram we have seen doublets of microtubules. So two microtubules lie side by side as a pair called as doublet in the cilia and flagellum structure. But here in the centriole, 3 microtubules lie side by side and forms as a triplet. Understand? So peripherally, how many triplets of microtubules with evenly spaced are present? 9. You can count them. They are exactly 9. Evenly spaced. Equal space can be seen between one triplet to another triplet. Location of these triplets are peripheral side. It means border side of the centriole. And these are made up of with a protein called tubulin. This is very important for NEET examination. So 9 peripheral evenly spaced microtubule triplets are present, made up of with tubulin protein. Next, one more point. Each of the peripheral fibril is a triplet, as we discussed just now. You can see on the screen very clearly, ma. The adjacent triplets are also linked. So 9 triplets are there, no? 9 triplets are also interconnected with each other. Understand? You can see very clearly in this diagram, 9 triplets are also interconnected with each other, and the central port, I mean every centriole has a central portion like cilia and flagella. The central part of the proximal region of the centriole is proteinaceous, and it is called as hub. It is called as hub. So the central portion of the centriole is a proteinaceous, and the proteinaceous portion of the centriole is called as hub, which is connected with the tubules of the peripheral triplets by radial spokes. So you can see in this diagram very clearly, every hub region has a spokes that are connected with the 9 peripheral triplets. Understand? So those spokes are also made up of with proteins. So the central space is called as hub, which is proteinous. Peripheral 9 triplets are also made up of with proteins, and the spokes are also made up of with proteins. Understood? Next, a very important function. Now let's talk. The centrioles form the basal body of cilia and flagella. So what is the role of cilia? Cilia plays a major role. It is nothing but formation of basal part of the cilia and flagella. And one more important, you can see on the screen, the spindle fibers that gives rise to spindle apparatus during the cell division in animal cells. So the centriole also helps to animal cells during cell division to divide the parent cell into 2 daughter cells by forming the spindle fibers. Those fibers are also made up of with a protein called tubulin, and these spindle fibers that gives rise to spindle apparatus. Those spindle apparatus are helping to the animal cells during cell division. Understand? So animal cells divide and produces daughter cells with the help of a important cell organelle. It is nothing but centriole. These centrioles are absent in plant cells, exclusively present in animal cells only. You can see centriole structure on the screen. They are present exclusively in animal cells. This concept is very important for competitive examinations like NEET and KCET. And these centrioles, as I told you, having a specifications, it is nothing but forming the basal body of the cilia and flagellum. Understand? So students, with this I conclude this today's video. Hope you understood. We will continue this chapter in the next video. Thank you one and all.